On this slide, you're looking at a little bit of a fungal culture that I grew for us here in my home. What you're looking at is a glass casserole dish in which I placed some canned pears. If you've ever purchased canned pears before in the grocery store, you know that they can come in heavy syrup or they can come just in pear juice. So I bought the kind that are in a heavy syrup to ensure that these pears were very, very sweet. All I did was I poured the can of pears into my casserole dish, and then I left the dish on the counter. And after a couple of days, this happened. What you're seeing growing on these pears are fungal species. And I've got a couple of different species growing. I can tell because I've got colonies growing that look a little bit different from one another. I can see different kinds of colony morphology here. I can see one type of colony that looks a greenish gray color, and I've got another type of colony growing that's a white color. I may have even other colonies growing. I may have one that's particularly small growing here. It's hard to tell this early on. But I've clearly got multiple types, multiple species of fungus growing on these pears. Now, the fact that this is fruit and it's very sweet is what allowed me to get fungus to grow on it without getting a lot of bacteria growing. Remember, bacteria don't like to grow in very sweet foods because the available water is too low for them. The sugar inside the food binds to the water molecules and makes it less available to the bacterial cells to use. Fungi have a lower requirement for available water, and so they grow just fine on sweet foods. Now, at this point, these colonies are relatively close to the surface of the pears. Some of the colonies are raised up, some of these are a little flatter towards the surface, but they're still in the colony form. In other words, there are few enough cells still that the fungus is growing simply as individual colonies. But I want you to notice this area right here. Do you see how there's a little bit of sort of a fuzzy cloud of white right here? This is a place where the fungus has started to grow in a slightly different form. The fungus has started to grow upwards off the surface of the pears, and it's in a more filamentous form. In other words, it's starting to grow as hyphae. I left the culture on the counter, and this is what happened. Now, I do want to let you know that I kept this covered because, as you can see, if I hadn't, my kitchen would be full of all kinds of fungal spores. So I kept this covered except for when I was taking pictures. This is the same culture a few days later. All of this white, cloudy looking, sort of puffy material, these are hyphae that have grown upwards off the surface of the pears. You can still see some colonies. You can still see particularly some of those green colonies. Here's some black growing. Over here I've got more black, I've got some white, I've got some green. Remember, it's likely I have multiple species growing. But what's most remarkable is all of this mold that's growing, and it's coming up off the surface. Remember, once it starts to come up off the surface, these hyphae become reproductive. They're going to start to create spores and send those spores into the environment. It's how the organisms reproduce. So if you look at these organisms, these colonies growing in the corner here, you can see that some of them are growing as black mold. And again, I don't want you to see black mold and immediately think that it's some horrible toxic mold. It's not. There are very few molds that grow black that produce harmful toxins. Most of them are perfectly harmless. So, the best way to examine these organisms is to get a sample of them and put them underneath my microscope. I put this picture in because I wanted you to see how I went about collecting this mold. If you try to take a sample of this fluffy white material, basically what'll happen is it will just collapse. It's so airy and lightweight and delicate. It's very, very difficult to get a sample of it. Notice what I'm doing. I have a gloved hand here, and I've got a piece of just regular cellophane tape in my hands. I've created a little circle out of the tape with the sticky side outward. And all you have to do is take that tape and gently touch it to the white hypha, to the mold that's growing up off the surface. And you'll collect enough of the cells to see underneath the microscope. This is a little trick that I learned after years and years of working in the microbe lab. Other people taught me this trick, but it's a very useful one to know. This image over here on the right, this is just some mold growing on Sabarodes agar. This is a silly little design that I made with the mold, but it's growing on that particular kind of culture media that encourages the growth of fungus while inhibiting the growth of bacteria. To finish up today, I wanted to talk about a clinical application in human medicine. Remember, the only microbial fungus, the only true fungal microbe is yeast. It's single-celled and it's microscopic. And the most common yeast that causes disease in humans is one called Candida. Over here on the left-hand side, you're looking at Sabarodes agar with Candida yeast growing on it. Remember, you cannot look at colonies growing on an agar plate and know for certain if you've got bacteria or you've got fungus. You have to take a sample and look at it underneath the microscope. And that's what's going on over here. We are underneath the 20x objective lens here. These cells that you're looking at are much too big to be bacteria. These are yeast cells. They've been stained so we can see them a little bit better. Notice that they're mostly sort of an oval shape or an egg shape, as some people describe them. The cells are different sizes. Because this is a budding yeast, some of the budding cells are just still young. They haven't grown to their full adult size yet. But this is this yeast that's called Candida. Now, there are different species of Candida, and some forms of Candida don't cause serious disease in us. This one, however, is of growing concern. This is a type of Candida called Candida Oris. And this organism has been in the news a lot lately because it causes an invasive fungal infection. Remember, we don't expect to see serious fungal disease in people who don't have other disease processes going on. We don't expect healthy people with functioning immune systems to come down with things like fungal pneumonia. And yet, in other parts of the world particularly, this Candida Oris yeast is causing that kind of a problem. So this is a pathogen that the Centers for Disease Control and the World Health Organization is keeping an eye on because it seems to be able to cause disease in otherwise healthy human beings. That's all I have for you today. The next time we get together, we're going to be talking about special media or specialized culture media that we use in the microbiology lab. When we talk about special media, we will also start conducting experiments together while we're in our Zoom labs. So I will see you next time.